I'm Jen Dykstra. I'm Melanie's advisor. Very happy to be so and continuing. First, I want to thank your committee. Mike Siegler, who's a scientist in residence at the Gulf of Maine Lab and former NOAA marine biologist, Easton White. Particularly Easton for introducing me to Melanie, and she was her mentor while during her first internship at the Shoals, or her only internship at the Shoals. And Michael Pallas, who is in the Department of Natural Resources, Earth Sciences. I'm sorry. And I should say that we all collectively helped Mel kind of shape her thesis. She certainly led the charge, and so it's been a really nice time working with you all. Melanie graduated with academic honors and scientific research honors from Vassar University. I'm not really sure what the difference is, but yeah, you can explain that to me. Among other kind of academic accolades that she received, they're too numerous to kind of detail here. She majored in earth science with a minor in mathematics. I first met Melanie at the Shoals Marine Laboratory when she was a fisheries intern for Easton. I poached her. Sorry, Easton. My first impressions of Melanie were enthusiasm, goal, and team-oriented, and those first impressions were spot on. She's been an invaluable member, certainly of my team, providing feedback during lab meetings, helping out other students, and again, her enthusiasm is just so contagious. Apart from her studies, she became a math tutor here at UNH for pre-calculus and calculus courses, and also participated in our outreach Ocean Discovery. And again, her enthusiasm for teaching others about her research was just positively contagious. Her enthusiasm, curiosity, and intellect made advising her a real pleasure, and I'm very happy to say she is staying on to do her PhD in natural resources.
Awesome. With that introduction, we're going to jump right in. I'm a little soggy today, but that's kind of fitting because we are going to be looking at how I've been predicting distribution patterns in nearshore fish and invertebrate communities. This will be about 45 minutes, and then we'll kind of have some time for questions.
Oh, and I will say I will ask her committee members to refrain from asking questions until after everyone has left the room.
Yep. So to follow along with the presentation, you can kind of keep track of where we're at with this little bar along the bottom. That'll kind of give you an overview. Also, if you have particular questions about a certain slide, feel free to jot down the number inside this little fish icon. That is the slide number. So we're going to start with the importance of the nearshore. In general, our nearshores are really important. So these are our coastal areas. What we see here is a figure showing us fishing effort from 2017. Now, what I want you to notice is these red areas are where we've got higher fishing effort. And in general, we are seeing higher fishing effort all along our coastlines. In addition, our nearshores are really ecologically important. Here we've got another figure. This shows us species richness. Again, we've got higher species richness in red, and in general, we see those areas with high species richness right in along our coastlines, also a lot over here, but along our coastlines in general. We can also kind of break down how species are using our nearshore areas. This is a figure showing us how 59 species advised by the International Council for the Exploration of the Sea are using these coastal habitats. In general, we see a lot of use in terms of nursery grounds, feeding grounds, and then also some use of these habitats for spawning and migration. Patterns like this have led a lot of our coastal areas to be designated as what we call essential fish habitat. This is defined by the Magnuson-Stevens Fisheries and Conservation Management Act as those waters and substrate necessary to fish for spawning, breeding, feeding, and growth to maturity. And then our last little broad kind of overview of the importance here is that these nearshore areas are very vulnerable to decline. So this figure is showing us where we have a lot of high human impacts and high species richness. So in red, what we're seeing is those areas with a high human impact score as well as high species richness. And again, we see a lot of those points concentrated along our coastlines. With all of this, I have identified 3 main problems in the near shore. I'll give you just those 3 main problems, and then we'll talk about them in a little bit more detail. The first main problem is the monitoring gap in the near shore. Our second problem is unknown species responses to terrain and habitat modification. And our third main problem is shifting species distributions. Let's talk about each of those in a bit more detail. We'll start with this monitoring gap in the near shore. This gap arises from the different types of ways we collect fisheries data. There are 3 main types: recreational data, commercial data, and scientific data. Those first 2 types are what we call fishery-dependent data. What this means is we've actually got fishermen going out, catching these fish, and recording what fish they've caught. This is really great because there is a lot of this data, but we're not recording where we don't catch fish, so we don't have zeros in that data set. That can be a problem because absence data is really important when we get to species distribution modeling. In addition, these types of data tend to be biased towards certain fishing sites, and they tend to be a bit lower resolution. Our scientific data is fishery independent. That's really nice because it allows us to standardize our sampling techniques and collect a lot of that absence data that we couldn't with our fishery dependent data. Trawling is one popular example, but trawling and a lot of our other common examples tend to be inappropriate for sampling shallow, structurally complex, or hard-bottomed habitats. And a lot of habitats that have high recreational and commercial use also pose some extra challenges. Because of this, there is a general monitoring gap in the nearshore. So what this means is we tend to not know where species are in our nearshore and what sites they're using. I aim to address this with my first objective, which was to characterize nearshore distributions of mobile marine fish and invertebrates. We're going to get into this second main problem with a little bit of an overview. Here we've got a figure that is actually from one of Jen's papers, and so we're just going to use this as kind of an example. What we see here over the past few decades is generally a shift from native species, shown in blue, so this is native seaweeds, towards more invasive species, shown in this kind of orangey color. And so we've been seeing a lot of these habitat shifts, as well as other things like shoreline modification and development. And this is a real problem because even though we know that these shifts are occurring, we don't really know how species are responding to those shifts. And so my second main objective was to assess the importance of predictor variables in nearshore species distribution modeling. Now, in addition to these habitat variables, I also did consider some of these bathymetrically derived variables, such as depth, ruggedness, slope, curvature, and different substrate types. The third main problem is shifting species distributions. So what we've generally been seeing is species moving northward, and then we don't see it in this particular figure, but we've seen patterns of species moving offshore towards deeper depths. We've observed this in 48 different commercially and ecologically important species all along our northeast shelf. And this has become a really big problem for fisheries management, because if we don't really know where these species are, it's really hard to keep track of them and manage them. Also, one of the main concerns is that we might see mismatches between predator and prey distributions. So if we've got a really important predator that we are fishing for commercial purposes, but that predator can't catch its prey, it's going to be a lot less productive than otherwise. One other concern that we've seen is novel bycatch risks. So that is a problem because we don't really know how these new combinations of species might impact our fishing. And so I tried to address this with my third objective, which was to understand species co-occurrence patterns. With all this being said, we're going to be taking a look at the Gulf of Maine, which is shown here. You might be able to make it out, but we're kind of somewhere right in there. That's where we're located today. And the Gulf of Maine is a particularly important system for studying these fisheries. Here we've got a figure showing us Gulf of Maine seafood landings since 1982. In black, we've got our total commercial landings, and then in our blue and kind of this pinkish color, we've got different breakdowns of those species. Essentially, what we've been seeing is a general average decline in our fisheries landings in the Gulf of Maine. Now, you might not be super familiar with this, but you may have heard about the collapse of the Gulf of Maine cod fishery, which is one of the most common examples of this. Now, I'm specifically going to be collecting data, or I have collected data, from the Isles of Shoals and the coastline of New Hampshire and Maine. So here's the Piscataqua Cove River, here's the Isle of Shoals. Something you might notice in this figure is that most of my data was collected around Appledore Island. And so most of my results are focused on talking about this data. I made a couple different comparisons between the other sites, but that's where we'll focus today. So before we get into that, we need to figure out how did I even look at what fish were using which sites. This is actually a really interesting question in the Gulf of Maine. Because here we've got 4 videos just within this one little cove here. And what we see is a lot of differences between these habitats. So in this bottom video, we have a lot of kelp. Up here, we've got somewhat of a drop-off, so it's overlooking this drop-off in the bedrock. Over here, we have a lot of boulders. And then over here, we've got some more turfy algae, and then kind of this undulating pattern in the bedrock. And so these are from these different sites over these fine spatial scales. And so what we generally observe in the Gulf of Maine and other people have observed is there is this high habitat heterogeneity over very fine spatial scales. This is a problem because if we were to introduce bait into this environment, we would risk attracting species from neighboring environments because the bait plume, especially in nearshore areas, is really unpredictable. So I decided to use an unbaited method, specifically remote underwater video systems, or RUVS. These are a really great method because they are a passive method. They minimize the disturbance to organisms, the disturbance to ecosystems. They also minimize our observer bias, as well as the deviation from home ranges. So the way these systems work, this is a little conceptual figure I made. We are going to deploy these cameras onto the seafloor, and then I left them to record for an hour at a time. They recorded continuous underwater footage, and then I went back and analyzed which fish I was seeing, and also invertebrates. I collected 197 hours of footage in total. When I was actually going through and analyzing the different individuals I saw, I grouped them into what we call functional guilds. Now to do this, I looked at a bunch of different papers. Don't worry about reading this. This is just kind of to show you all the literature that's out there, or just some examples of that. But essentially, functional guilds are groups of species that use resources in similar ways. And so I looked at some papers that have grouped these species into these guilds in the past. I also analyzed the different species' diets and their habits and behaviors to try and group them together. Some of the functional guilds did include just one species, and so those are species that occupied a distinct ecological niche, whereas other guilds occupied multiple niches. And so those guilds had more species. To get the actual numbers, we have an interesting thing that we need to address. So in this video, we see a pollock come in from the right side of the video. It swims it off on the left, and then what we're going to see is a pollock swimming in on the left right here. We can kind of use our deduction and figure out that's probably the same pollock swimming in and out of the same frame of the video. And so we don't want to count that as 2 pollock because that was overcounted. Instead, we only count when we can see both 2 pollock on the screen at one time because we know for certain those are unique individuals. This allows us to avoid overcounting individuals, and this is a really common counting metric among underwater video systems. And so this is called the MaxN counting metric, so I'll be referring to that. We also call this a fish density metric because this is essentially the number of fish per video that we observed. So that kind of gives you an overview of how I collected this data. Now we're going to get into my objectives. My first objective was to characterize nearshore distributions of mobile marine fish and invertebrates. To start, we're going to just kind of get an overview of the different guilds that I saw. Here are 11 of the most common guilds I observed, and over here we've got their percent frequency in the different videos. What I noticed were that pollock and cunner were definitely the 2 guilds that were appearing in the most number of videos, so they appeared with the highest frequency. And so I'm going to talk mostly about these 2 guilds. Also, these 2 guilds are just single species, so pollock is a species and cunner is a species. So we'll be talking about those 2. The other ones I'll mention a little bit briefly, but we don't really have time to get into all of the different guilds today. This is a breakdown of all of my MaxN counts. And so this is comparing the coastline to the Isles of Shoals. The different colors are representing the different guilds. And so I really want to see what proportion of these MaxN counts were made up by our different guilds. Over along the coastline, we saw a higher proportion made up of schooling planktivores, such as mackerel and menhaden. Whereas along the Isles of Shoals, we saw a much larger proportion made up of cunner and pollock. I also looked at finer scale site differences. So here we've got a breakdown by site. And again, I did see some differences between our sites. So for example, over here, we've got a lot of school and planktivores. These sites had more cunner and invertebrates. Up here, we've got some more pollock in these ones. And then I also saw some differences out at the Isles of Shoals. So over here, we've got a lot more pollock, over here, a bit more cunner, and then some other variations. So I'll just kind of summarize my first objective here. I first observed differences between island and coastal communities. This has been observed in other places around the world and over larger scales in the Gulf of Maine, such as over hundreds of kilometers. But what I was observing suggested that this is also true over finer scales in the Gulf of Maine. I also observed differences in adjacent site utilization, which contributes to the ideas of seascape connectivity and differing spatial preferences among guilds. So I'm going to get into this one a little bit more with my second objective. So my second objective, again, was to assess the importance of predictor variables in nearshore species distribution modeling. Now, as you can kind of tell from this little slider along here, this is kind of like the big chunk of my thesis. And so I'm going to break this down into two parts to help you understand it a bit better. We'll start with the initial assumptions. So essentially, we're going to build two models that both assume different things about the importance of environmental or seafloor variables. And then we're going to actually dive in and figure out the individual importances of each of those variables. All right, I started with something a bit unique. So I really wanted to capture this idea where if you're out fishing and you catch a lot of fish, you might keep fishing in similar areas because you might expect those areas might also have a lot of fish. Whereas if you're out fishing and you catch no fish, you might think, oh, we're not going to catch a lot of fish here, so you might decide you might want to move on and fish somewhere else. And so I want to capture that, so I used inverse distance weighting. For this, we start with the idea that we're trying to interpolate our fish densities right here. So essentially the idea is we don't have a data point out of that red point. We want to figure out if we dropped an underwater camera there, what density of fish would we expect to see? We do that by looking at neighboring points. Those points are going to have a stronger influence on affecting how we interpolate that density at the red point than points further away. But some of you might be thinking, this point right here is on the other side of this landmass. And this is just kind of an example. This is not what my study site looked like. It's just from a paper that talks about inverse path distance weighting. So this point right here, we actually have probably further away because if you were a fish, you'd have to go all the way around that landmass. And so if you're thinking that, I would say, yeah, that's probably right. So what we do is we take into account this aspect. And so this is called inverse path distance weighting. The idea here is the points that are closer in terms of the path are going to have stronger influence on the interpolation, whereas points that are further away by the path to get to that point around barrier features will have less of an influence. So again, that's inverse path distance weighting. The other model I used was random forest models. I chose random forest models as my model that takes into account the importance of predictor variables because they're capable of considering many predictor variables even if we have a relatively small data set. Also, they avoid assuming the strength or shape of associations between our response and predictors, and they lend themselves well to calculating variable inferences, which was really great because that is the point of this objective. To understand random forests, we're going to start with this idea of a piecewise function. And if you don't really like math, don't worry, we're going to break it down in kind of a simple way. So what we're going to do is we're going to consider one of our predictors. For example, the percentage cover of kelp beds. And then we're going to consider a condition. Now, I just chose some examples. The random forest is actually going to go through and figure out these conditions and our predictions, but these are just kind of examples. Essentially, we check, is the percentage cover of kelp less than 75%? If yes, we might predict there are 0 pollock there, for example. Whereas if we have percentage cover of kelp greater than or equal to 75%, we might predict, for example, 5 pollock are at that site. Now we're going to take this idea of a piecewise function and expand it into a decision tree. This is the foundation of a random forest. So again, the random forest is going to figure this all out for us, but this is an example. We take an environmental variable and pass it down this decision tree until we get to our first split. The random forest is going to figure out which variable it's going to consider, for example, percent cover of kelp, and it's going to compare it against the cutoff, for example, 75%. It's going to check if we have a percent cover less than 75%, we'll pass it down the yes branch, and we might predict 0 pollen. If we pick another variable or another set of environmental variables, pass it down the same tree, we might check, okay, is percent cover less than 75%? In this case, we might say no, so we go down this branch. Now we're going to check a different variable. For example, is the depth deeper than 12 meters below mean low or low water? If yes, we'll pass down this branch, and we might predict 8 pollock. Random forests are going to take this idea and build many decision trees. Each tree is built using a random subset of the data, so we sample with replacement to get our data. We then train our trees, and the random forest is going to figure out which variables and what cutoffs we want to use. And we also further decorrelate these trees by at each split, we're going to consider a random subset of our predictor variables. So we have differences between our trees. What this means is that if we pass the same set of environmental characteristics down each tree, we're probably going to get different predictions. So to figure out our overall mean predicted density of fish, we're going to average those and get 7 pollock per video as an example. Now we call this a random forest because these kind of look like the branches of a tree. If we flip that, we've got now a forest. We only considered 3 trees here, but we usually consider many, many more, such as 500 trees. So that's what I use. But when we make these random forests, there's a lot of decisions that we need to make. For example, should we perform data transformations? How are we going to handle outliers? What are our outlier cutoffs? And how many variables are we going to consider at each split in the tree? So there's a problem. There are millions of possible models. The solution for me was to use a genetic algorithm. This is kind of like if you're trying to make the best possible bowl of soup. Essentially, we need to figure out what ingredients to use and how many of each ingredient we want to use. I use a genetic algorithm to figure this out. So, genetic algorithms are really cool. I'm really excited about them. So if you have questions later, feel free to ask me. So essentially, we're going to start with a model which we consider an individual. We're going to use 4 ingredients, the ones that I said earlier, but usually we consider a lot more. And we're going to essentially choose some inputs for those ingredients. For example, a log x plus 1 transformation, we might decide that, yep, we want to exclude outliers. We might choose an outlier cutoff of 1.5 times the interquartile range, and we might use an mtri of 6 random variables. We're going to start by numerically encoding this into real numbers, and we consider this the DNA of our individual. So the idea here is we're going to essentially apply the idea of evolution and natural selection to choose the best possible model without having to check every single one. So we've got our model with our DNA. In the first iteration of the genetic algorithm, we generate a series of models that each has unique DNA. What that means is we've got different inputs for each of our variables, so our hyperparameters, we like to call them. So for example, we've got different transformation, true or false, we've got different outlier cutoffs, and we've got different mtri values. So we've got different combinations of our genes, so to speak. We now assess the fitness. How likely are these models to survive to create our next generation? Our fitness is determined by checking the error rates when we run our random forest, and we want lower error rates. So these models that have the lower error rates are going to survive to create our next generation. To create the next generation, we take the models that survive and we perform something called arithmetic crossover. This is going to blend our favorable genes so that we get new DNA. And so this is our new DNA, which allows us to make a new set of models. We again assess the fitness by calculating our error rates and determine which models have the best error rates, so the lower error rates. And we keep repeating this process until eventually we get just one model and we no longer see improvement between our generations. And so this kind of gives us our set of ingredients that we're going to use to make that soup. I applied this process to both my inverse path distance weighting and my random forests. The other thing that I want to talk about before getting into the results is how I handled the test and validation data sets. So we start with this idea that we're going to take our data set and divide it into 5 equal partitions. I set aside 20% of the data as the test data. This is really important because this is essentially what we use to assess the performance of the model when it sees data that it has never seen before. So we don't use this test data to train or tune the model at all. We're going to take this other set of data up here, and what we're going to do is duplicate it, so we've got 4 copies. This is so that we can apply k-fold cross-validation, which is common in model building. What we're going to do is we're going to create 4 identical models. The differences between these models is we're using a different partition of our data set as our validation data for each model. The idea here is that it strengthens our models because in general, when we look at the set or suite of our 4 models, we've got models that have all seen the training, all of our data as both training and validation data. So it just strengthens our models. So that's our validation data. And then to calculate my validation test errors, I use mean squared error, which is shown here. If you don't want to worry about the math, you can just take my word. We want lower mean squared error. And then you'll also see root mean squared error, which is just the square root of mean squared error. And again, we want lower root mean squared error. All right, so getting into some of these results, just to orient you, we've got our IPDW models over here. Again, that's kind of based off of the idea that we are interpolating those fish densities in our invertebrate densities. Over here, we've got our random forest model, so that's the one that's going to take into account our environmental and seafloor characteristics. The first thing I noticed, so this is comparing the predicted densities of cunner. I saw that there were some similarities in areas where we had high predicted mean densities of cunner, but I also did definitely notice some differences. So I compared the error rates, and I found that in general, the random forest model had lower error rates, so it was performing a bit better. Here's our IPDW for pollock now, and our random forest for pollock. And again, I did observe some similarities in areas where we had high predicted mean densities, but also some differences. And in general, the random forest models tended to perform a bit better. I also just wanted to throw up our little cunner random forest and our pollock random forest right here. And so these just kind of comparing where we had high predicted mean densities for both of our species. So cunner are shown in red, pollock in white. And so we did see some overlaps between areas where we had high predicted mean densities, but there were also definitely some differences. This is a summary figure showing our error rates for our different guilds. So here we've got root mean squared error on our y-axis and our different guilds on the x-axis. Circled in red is our IPDW model, so that's the error rate for the IPDW. So in general, this is showing us that our IPDW model tended to perform a bit worse than our sets of random forest models. So these are different random forest models that I ran. In general, I did observe this across all the guilds. And then here we've got our test errors. So again, in general, we did see our IPDW models performing a bit worse, so they had higher error rates, but this had a bit more variability because our test data, remember, was only 20% of the data, whereas the validation data, we really strengthened that using that k-fold cross-validation method. All right, to summarize this first part of Objective 2, we have considered environmental and seafloor variables, and we've seen that those random forest models, which take into account these variables, did perform a bit better than the IPDW models. In general, I found that the IPDW models, or the inverse path distance weighting models, gave us a kind of this nice, easy overview of our data set, but the random forest resulted in better predictions. So this supports the assertions of the importance of site and environmental variables for predicting fish and mobile marine species distributions. Getting into the second part of my second objective, we're going to talk about variable importances. We're going to start by assuming that there is a linear relationship between pollock densities and longitude. Now, the actual relationship is a lot more complicated than this. We're just going to assume this for the purpose of explanation. What we're going to do is pass this initial data set through our random forest and calculate an initial baseline mean squared error. Next, we are going to run a permutation and essentially break that relationship between the longitude and pollock densities. This I performed 200 different times because this is a random procedure, so if I didn't, then there would be a bit of uncertainty. So I did perform it 200 times to kind of stabilize these results a bit more. We pass this new data set through our random forest again and figure out a new mean squared error. Now, the idea here is that if pollock densities have a strong relationship with longitude, if longitude is a really important predictor, breaking this relationship is going to result in a greater increase in mean squared error, because without that variable, the model will perform worse. If that doesn't make a lot of sense, you can kind of think about our bowl of soup. So say we've got this tomato soup. If we take out one of the really important ingredients, such as the tomato, it's probably going to taste a lot worse than it originally did. The same idea we apply here. So we take out an important variable, our model is going to perform worse, so we have a greater increase in percent increase in mean squared error. Meanwhile, if we've got a variable that isn't as important for our predictions, we might see less of an increase in mean squared error. This is kind of like if you take your tomato soup and you took out a less important ingredient, such as the pepper. Will it taste a little bit worse? Yeah, it might taste a little bit worse, but it's still going to be roughly the same soup. This is the idea that I used to create this little figure over here, and this is a very common strategy among random forest models. So this percent increase in mean squared error is very common. Essentially, to orient you on this figure, we've got our environmental and seafloor variables here, and we've got our guilds along our columns. Our colors are representing our percent increase in mean squared error. So red are more important predictors, and blue are less important. Also, I put our little importance ranks inside each square. So 1 is the most important predictor for a given guild, whereas 11 would be the least important predictor. So what did I find? Well, I found that kelp bed was the most important predictor for cunner, and I found that latitude and longitude were the most important predictors for pollock. In general, I found that our geographic position variables were important for 3 of our guilds, the most important predictor. I found that our terrain variables derived from bathymetry were most important for 3 guilds as well, so 27%. And I found that our habitat or vegetation variables were important for 5 of the guilds, so about 45% of the guilds. We can also look at how each of these predictors actually impacts our predictions. And I'm not going to talk about every single part of this figure or each individual guild, but we can just kind of look at one of these as an example. So this is called an accumulated local effects plot for cunner. Essentially what we've got is our predictor variable along this x-axis. So here we've got percentage cover of kelp beds. And then along our y-axis, this is the change in our predicted density of cunner. So the 0 line is going to represent our average prediction of cunner. As we have more cover of kelp, we're going to see on average higher predictions of cunner. So for example, if our average prediction of cunner is 3 cunner, if we've got something like 75% cover of kelp, we might see a prediction more like 3.75 cunner per video on average. So to summarize the findings from this objective, part 2, essentially I found that kelp was the most important predictor for cunner, and this makes a lot of sense. We know that cunner use kelp for refuge, so we see them a lot hiding under the blades of the kelp. And this is a cause for concern because in general, we have seen an average decline in kelp in the Gulf of Maine and around the world as a whole. And cunner are a really ecologically important species. They occupy the middle of our food chain, so they're an important predator species and an important prey species. I also found that geographic position was the most important predictor for pollock. This actually makes a lot of sense because this is kind of suggesting that pollock are less dependent on fine-scale seascape attributes. And we know that pollock are a semi-pelagic species that go offshore more often. And so they're probably more dependent on these large-scale oceanographic variables. I also found that variable importances varied between guilds in general. And so this is suggesting that different guilds are going to respond to habitat and terrain modification in different ways. All right, with that, we're going to get into the third and final objective, which was to understand species co-occurrence patterns. So I want to start by bringing your attention to this figure. So essentially what we're looking at here is Spearman rank correlation coefficients. Now, if you don't want to worry about the Spearman rank part, you can kind of think of these as correlation coefficients. Values close to 1 indicate a stronger relationship between our 2 guilds, and values close to -1 represent a stronger negative correlation. To read this, we're going to look at a particular row and a particular column. And so within the box is our correlation coefficient for that pair of guilds. So for example, between crabs and lobster, I observed a correlation coefficient of about 0.65. Now to calculate these correlation coefficients, I took the random forest maps and essentially figured out the correlations of the predicted mean densities. Between seals and pollock, I observed a correlation coefficient of 0.67. So this was really interesting to kind of get an overview of what our random forest models were telling us about co-occurrence patterns. However, I noticed that cunner and pollock had a really low correlation coefficient. So it was 0.12, which is pretty close to 0. Now that's against expectations because cunner and pollock were the 2 most common guilds seen in the study, as you might remember from that first objective. So what I realized was happening is that with these correlation coefficients, we're really closely modeling the actual MaxN densities, and we're weighting these 0 counts the same. So I wanted to kind of take a step back and kind of look at the broad picture and essentially summarize these distributions and figure out overall, what are we seeing in terms of co-occurrence and distribution overlaps from a broad lens. I started by using something called standard deviational ellipses. This is a common method in spatial studies, and the idea here is we're going to take all of our sample points and find the center of the distribution. So essentially, if you had these points balanced on, say, a little slate, where would you be able to balance all of those points on a single point? And so that's what the star represents. That's the distribution center. We then figure out what is one standard deviation away from that mean center, and so that's what this represents. In essence, what you see here is what we would expect to see if a particular gilt was evenly distributed around the entire island. What we actually saw were differences in this. So here we've got cunner shown in yellow and pollock shown in blue. And so what we see is some differences from this baseline assumption. So cunner were pretty close to being evenly distributed all around the island, whereas pollock were showing some more spatial preferences. The theory here is that if we had gilt distributions that showed a lot of similarities, if we had some overlaps between their distributions, we would expect to see a greater percentage overlap in their distributions. For example, with pollock and cunner, we saw an overlap of 59.61%. I also came up with this other method, and this is another common method in spatial studies called concave hulls. The idea here is that we make a little envelope along all of our sample points. And so essentially what I was doing with the individual gilts is I created this concave hull around all the points where we had a presence of a particular gilt. For example, for the pollock one, again, the colors are the same, so blue for pollock, yellow for cunner. For the pollock concave hull, what we see is pollock were generally absent from the south side of the island. And so we're really just trying to summarize these distributions with some really broad scale pictures. And the idea again is if we got a lot of overlap between their distributions, we're going to see higher percent overlaps. So for pollock and cunner, I saw a percent overlap of 59.46%. Now, both of these methods are making some assumptions. So to get a kind of broad picture that really stabilizes our percent overlap analysis, I took an average of these two. That's what we're going to see in this figure. So here we've got our pollock and cunner percent overlap from these analyses. And here's the full figure. So I want to point out that this looks a lot like the Spearman rank correlation figure, but we read it a little bit different. So all these values are representing our percent overlaps, not our Spearman rank correlations. We read it the same way, where we look at a particular row in a particular column, and we use that to find the pairwise percent overlap. So for example, I saw high percent overlap between crabs and cunner, so 61.55%. I saw high percent overlap between crabs and lobster, 53.84%, and high percent overlap between seabirds and cunner, so 52.71%. So I took a step back from this as well, and I also ran a cluster analysis. We're not going to talk too much about this analysis, but the idea here is we took all of our videos and we assessed the different communities in the videos. We then identified common community structures, so each of the colors here represents a different community, and each of these little points at the very bottom of the cluster analysis represents an individual video. I did this because this is a common way to assess our communities and kind of figure out which species or gilts are occurring together. We do that with an indicator species analysis, so I figured out the indicator gilts for each of the clusters, and then I compared that against the different methods that I used here in this objective. With that, I'm going to go through and summarize this objective. So I identified overlaps between cunner and crabs, cunner and seabirds, crabs and lobster, and pollock and seals. And so these are all representing predator and prey overlaps. Also, I do want to note that pollock and seals are a particular one to pay attention to because this does represent a potential bycatch risk. The Northeast Sink gillnet fishery is one of the fisheries that targets pollock. And between 2017 and 2021, we saw an annual average of about 1,300 gray seal mortalities attributed to this fishery. And between 2015 and 2019, we saw an annual average of about 300 harbor seal mortalities attributed to the Northeast Sink gillnet fishery. Among U.S. commercial fisheries, this was the leading cause of gray seal and harbor seal mortalities, so it's just something to keep in mind. With all of this, I am now just going to wrap up with a little bit of a conclusion. So I want to take you back to what we talked about in the introduction, which was the monitoring gap in the nearshore. Here we've got different points representing 81 different species. So this is biomass for those 81 different species. These are collected from the spring and fall tows between 2010 and 2019 by the Northeast Fisheries Science Center. Yeah. So what I want to bring your attention to is this monitoring gap in the nearshore, so we don't see a lot of points here. So what I really tried to do was to supplement the existing data by collecting some points in this scat. I also collected over a much finer spatial scale, so I'm really hoping that this can kind of add to the existing data sets by providing a new lens. In general, for my first problem, I addressed this with objective 1, which was characterizing nearshore distributions of mobile marine fish and invertebrates. And my key findings were that there were differences between island and coastal communities, and I also observed differences over fine spatial scales. In general, I'm really hoping that these findings can serve as a baseline for future monitoring and vulnerability assessments. For the second problem, I addressed this with objective 2, which was to assess the importance of predictor variables in nearshore species distribution modeling. My key findings were that the consideration of environmental and seafloor variables improved our predictions. So those random forest models tended to improve our predictions over the IPDW models. I also found that variable importances varied between guilds. And so I'm really hoping this can support the application of seafloor characteristics to mobile species distribution modeling in the nearshore. For my third and final problem, my goal was to understand species co-occurrence patterns. And my key findings were identifying predator and prey overlaps, and then also a potential bycatch risk between pollock and sliss. These are really meant to address some of those concerns regarding shifting species distributions, especially those pertaining to bycatch risks and mismatches between predator and prey species. So just kind of an overall broad summary. Based on problem 1, I have now described some current fine-scale distributions of commercially and ecologically important species. For problem 2, I have explored the relative importance of variables in predicting those distributions. Problem 3, I have identified some of those current co-occurrences and those overlaps between their distributions. And then in general, I've really compared some of these different methods and statistical analyses for their application to nearshore fish and invertebrate modeling. With that, I want to make a couple of different acknowledgements. And I first want to start by acknowledging the National Oceanic and Atmospheric Administration Award, which funded my research assistantship. So that's an award to the Center for Coastal and Ocean Mapping and Joint Hydrographic Center. I also received additional funding from the New Hampshire Sea Grant Development Funds, as well as the Karen Von Daum Research Fund. And I want to give a special thank you to a couple different people. So definitely my thesis committee. This definitely would not have been possible without all the feedback they gave me, all the answers to all the different questions that I asked. So that was incredibly helpful. It would not have been possible without them. Another big thank you to Madeline Wanger, who collected the data from 2023. All that RUVS data was collected by her. Big thank you to Shoals Marine Laboratory staff and facilities for helping me with this research and making this possible. A big thank you to the 2022 and 2023 summer undergraduate research groups. They helped with a lot of the data collection when I was out at the island and also when Madeline was out there. Big thank you to Connie. She came out on the boat with me. She drove the boat. It was great. She hung out with me when the videos were, down there on the seafloor. Also, she answered so many of my questions about random forests and also ArcGIS. So huge thank you to Connie. Big thank you to Glenna. They also answered a lot of my questions about ArcGIS. They've also been supporting me throughout this whole master's and really been by my side. Big thank you to Sandy for coming out on the boat and helping me with some of that data collection. Huge thank you to Sanjana. She's literally been here with me since day one of my master's thesis. Also, if that soup analogy helped you understand things, that was her idea, so you can thank her. But yeah, she's really been really important to me. She also came out on the boat and helped me collect a lot of the data and honestly has been my best friend and honestly family at this point. So big thank you to Sanjana. Also, big thank you to Anna and Julia from Easton's lab. They really got me oriented. They were some of the first graduate students I met here at UNH when I first became the fisheries undergraduate researcher. And so they introduced me to these remote underwater video systems and really got me started there. Big thank you to Brett as well, who I don't know if Brett is here, but Brett is also in the lab and also gave a lot of good feedback for a lot of my work. Another big thank you to Paul Johnson and Larry Ward. They also answered a lot of my questions about data and how to apply that data. And a big thank you to all of my family and friends, everyone who I wasn't able to thank individually. I promise a lot of you are in my actual written thesis, so don't worry. But also a big thank you to the family that's here and everyone who's on Zoom. Thank you to all of them. And in general, thank you to everyone who's here today. And with that, I am happy to answer any questions. Thanks.
the like variability in depth that you deploy the cameras at? Is it possible that the species you saw is biased by what depth the camera was at?
Yes, absolutely. So the depths that I was looking at were pretty much focused between 0 and 15 meters below mean lower low water. So all of these species and guild are definitely more common. The ones that are considered the rare guild in the study are things like cod and then also some sculpins and sea ravens. Those are more common at deeper depths. So those are definitely rare in terms of rare because they weren't seen as much in this study, but not necessarily rare because they're not in the Gulf of Maine, just like in the depths that I was looking. So yes, definitely. Yeah.
With the time of day that you collected your footage, do you have any consideration as to like, I don't know, how long the sun had been up, what tidal cycle you're in, or was it just more like vessel of opportunity? We have the time. Like, how did you pick your time of day to do your deployment?
That is a fantastic question. So a lot of the points from 2022 varied in the time of day. So what I did was I considered time of day as one of the variables. So I didn't have them in the maps because that doesn't lend itself as well to mapping. But for one of those figures where I showed the error rate for random forests, one of the variables was the time of day. I also considered the day of the season and a couple other I considered the year and a couple other like time related variables. I didn't see any strong associations because in general they were pretty much focused in this kind of same window, kind of focusing on the 9 to 10 a.m. window. And that was chosen because it just improves a lot of our visible clarity when we have these underwater videos. But we do know that species definitely are more active at different times of the day. It just helped that most of the videos were pretty much focused on this one window. But yeah, I did consider it to see if there were any strong patterns. Yeah.
I'm not sure if you'd already mentioned it, but when did you start taking those videos? Like, what was the duration of time?
Yep, so it ranged from May to August for the most part. So it was the summer season, and then a couple from September. Yeah. Owen?
Just gonna throw this out there, we don't have any questions from chat yet, but you have a whole flood of people saying congratulations, great job.
Aw, thank you guys. You guys are the best. That's so nice, yeah.
When you're dealing with biological populations, farming populations, you used inverse distance weighting on that. And so the assumption is that if I have 1,000 here and I have 100 here, then halfway between them, I've got halfway those two values. The reality is with certain biological populations, they don't do that, they clump. So some are territorial, some are more dispersed. In this, is there a need to incorporate that sort of thing, that locusts behave very differently than highly territorial animals, for example? Is there a need to do that? Is there a way to do that in what you've done?
That is a fabulous question. So that's actually something I considered a lot. So that is very true. Inverse path distance weighting is more commonly used for like environmental variables, such as interpolating like sea surface temperatures, and not so much for these community data. So the reason that I ended up choosing it is because a lot of our species were kind of prevalent everywhere. So it was mostly so that I could look at pollock and cunner, and because I wanted to explore that. So some of our species definitely do show territory and also preference towards these sites like I found. So it's really just a way to figure out, can we identify some of these hotspots using this method? Because I also tried some of the other methods like kernel density. I tried some of those other methods. They honestly did not work super well for representing what I was seeing. The one that got the closest to kind of representing this was the inverse path distance weighting. And I think it's mostly because it's coming from this idea where if we're looking at one of these gills that's really prevalent everywhere, we can kind of assume that we might see some patterns that we could interpolate. But like I was saying, it doesn't work as well, and it's probably coming from a lot of that idea of the sense it's biological data, so it doesn't work as well as some of these other methods for sure. But it's just something that I kind of wanted to explore because it's not really commonly applied. Yeah. Yeah, Sylvan.
I was curious about this slide 34.
Yeah.
When showed a
Yeah, there's a lot of slides. Hang on. You feel free to start asking if you want. Yeah. There we go. That one?
Yes, thank you. I was curious about essentially the graphic on the right where you have MPD values both within the survey area and also outside of it. And I was wondering how the outside values are determined if it's not necessarily if it's not a place that you had fish data.
Yeah, sorry, so you're asking about this black line, is that what you're asking?
Or the data outside the, like the MPD values outside of the black line and how that.
Yes, yes, so that black, yes, so the benefit of random forest is we can do some of this extrapolation. So because we're looking at these environmental characteristics, we can kind of guess what we might see outside of our sampled area. The black line though, so that is just matching where we had our sample site, so that's the concave hull around all of our sample points. And so that's really comparing just these two. So when I did comparisons of the random forest and the IPDW, I really compared just within the same spatial area. So that's more for visualization, showing where that was on fixed figures. Does that make sense? Does that answer your question?
I guess I'm just really curious about how much confidence you think you can have in the data, in the MPD value outside of the parameters, not like the model was derived based on those treatments.
That's a good question. I would have pretty good confidence kind of close to it. I think as we get to different depths, my confidence would drastically go down. Because as we get to those deeper depths, we do start seeing different species and different guilds. So I would say in similar depths, it's probably kind of similar and we can kind of extrapolate a little bit. But I would say as you get to those deeper depths. Yeah, definitely.
How did you decide on the sites that you were putting the cameras?
Yeah, so I looked at a couple different things. It started by looking at the intertidal transects, which is something around Appledore Island. So Appledore Island was chosen because that's where I first started as a fisheries intern. But it's also really interesting because there are these historic data sets. So there's these transects all around the island. So I chose sites. So you might kind of see like this one is kind of along this line. That is one of the intertidal transects, or I think that should, like a lot of these lines should kind of match up with the intertidal transects. And the idea there is hopefully it can be compared to some of those data sets. Also, so Madeline chose a couple different sites, and I think a lot of that was just trying to capture different habitats. So because I was doing some of these habitat comparisons, I wanted to get a range of kelp habitats, bear habitats, turfy algae habitats. So it was just kind of a couple different factors. Yeah. Yeah. Yeah, Tom.
Interesting from my perspective, because I'm used to dealing with, especially with your data swimming in and out of your field of. I mean, it seems like if you're pointed in one direction, you don't see anything that's going on behind you.
Yep.
And maybe in an area of uniform habitat or substrate, maybe that wouldn't matter. But if you're in a high habitat diversity area, does the point direction that you're pointing matter? Or have they considered, or have you considered, I know we talked about this briefly, multiple cameras around. So you don't have to worry about a fish swimming in and out, you see everything.
Yes.
And you don't have to worry about MaxN counts and things like that, you just get the counts, whatever that is.
I really want to employ that. So I've heard a lot about these new systems that are using 360-degree cameras, and that does resolve a lot of the questions, but there are still like the need for MaxN because fish will kind of swim away and then back in, and like when they swim further away, you can't see it. Yeah.
They're coming in and out, actually around behind.
Yeah.
So you only eliminate, even a buggy,
Right. Yeah, but in terms of like kind of which direction the camera is facing, so that we kind of think about it like the sample site is the area that we see. And so that's the fish that we're going to capture, the ones that are in that frame of the video. And so that's kind of that. Yeah.
In regards to RUVS, why does it have an nose? Like the little PVC pipe that, like, is that for that depth perception sort of question?
So,
or, yeah.
It started by the RUVS. You can attach bait to them to make them baited remote underwater video systems, or RUVS. And so you attach the bait cage and then attract fish that way. I ended up leaving them on for the remote underwater video systems because I wanted to keep things consistent. So I actually had to create a new iteration of them in 2024, I think, for that. Same structure, same dimensions. And I ended up keeping that arm even though we weren't using bait, because what I noticed were some of the species would actually rub against the arm. So some species are actually attracted to structure. And so I want to leave it pretty much the same, even though we weren't using bait, just to like keep everything consistent. Yeah. Yeah.
I was also curious about the like MaxN choice, because it seems like I understand how that prevents problems with overcounting. Does that also present problems with undercounting? Like, and I guess I'm just curious if there's a reason why that's less of a concern, or like if there's some way that that's accounted for.
Yes, yeah. So the MaxN is most popular one because it is an underestimate. And the reason for that is because it's like this biological, ecological data. If it's going to then be interpreted later on by someone else looking at the study, and someone might say, hey, we have so many pollock here, let's not worry about it. It's worse if we overestimate than if we underestimate. Because when it comes to conservation or things like that, it's much better to have an underestimate than an overestimate. Yeah, and we also, it's also nice because we kind of know more about the data points. Because if we were to count every single fish we see, some videos that would be all unique individuals. Other videos, it would be the same individual coming in and out of the frame. So it kind of just gives us some more consistency in how we interpret those actual numbers, if that makes sense.
As a non-biology person, will you go to slide 42?
42, yes, absolutely. Hang on, again, loading, buffering, okay, yeah.
So here you're talking about, you pointed out a couple species that had specifically latitude and longitude correlation, right? What do you suspect draws those species to latitude and longitude? So it could be a migratory thing, could it be they prefer a certain temperature? What's your hypothesis?
So there's this thing, there's this kind of characteristic of Appledore Island where each quadrant is a little bit different. And so this has been discussed in a paper that Jen actually was on and Jarrett Burns. And so each quadrant is kind of a little bit different. And so in the north side of the island, we've got these really steep slopes. And something we've observed at other islands around the Isles of Shoals is that when you've got these steep slopes in the upstream side of the prevailing current, you might get some upwelling. So it's been hypothesized a lot that there might be some upwelling around the Isles of Shoals, which could promote productivity and provide some more food sources. So pollock might be having, more food available in certain areas. So kind of when we talk about these geographic position variables, we're kind of thinking about this broad scale. So if you think back to the random forest, we really saw pollock really abundant on the east side of the island. So it's kind of this broad scale. That's the exposed side of the island. We've got more wave action. It's just broader oceanographic conditions. Whereas the west side is a bit more protected. It faces the mainland. So things like cunner, they might be more likely to be over there than on the broad open side because there's less wave action, that kind of thing. Yeah. Yeah.
How did you position the RUVS on the seafloor?
Good question. So I pretty much just lowered them. It sounds simple, but it's essentially the RUVS are these like PVC structures. They've got weights at the bottom. You just kind of take the structure and you kind of just gently toss it off the side of the boat, and then you just kind of feed it some lime and let it sink to the bottom. And it kind of, so the other benefit of that arm is it kind of helps it land upright in a lot of cases. So if it like initially lands and it's kind of like tilted, the arm is going to prevent it from kind of flipping, so it kind of flips back. There were definitely some RUVS that kind of landed sideways, upside down. I just completely cut those out of analysis, so those are not in the analysis. But yeah, they generally landed pretty nice.
Was the assumption that their position was the position of the vessel? You already had that?
Yeah.
Or also a few times the arms got stuck in the lumps as
True.
drive the boat in circles too.
Yeah, it was really hard to pull up sometimes. My arms were really tired trying to pull, because I'm essentially pulling the boat along.
How many RUVS are still the boats?
No. We got them all back. They're expensive cameras. Yeah. Don't want to lose this.
We have time for maybe one or two more questions.
Yeah.
Do you have recommendations to update the next generation of RUVS? If you had a wish list for
Yes,
what to put on it?
that is such a good question. I really want to put on some, if I were to do this study again, I would totally put on some sensors that would figure out what is the bottom temperature. Things like maybe salinity or dissolved oxygen, just some of those environmental characteristics at the bottom. That would be so great. And so this year, there actually is an intern out at the Isles of Shoals who's deploying RUVS, so a new set of RUVS. And at each point, actually doing this CTD task to figure out some of those variables. So that's one of the main updates. Also, I think it'd be really nice to add a little PVC cage around the camera, just to make sure that when it gets pulled up, if the RUVS tips, it doesn't, scrape the camera along the seafloor. There were a couple times that it maybe dragged through some sand, and I had to really hose it down, but yeah. So I might do that. Yeah.
Do you plan to tie any of this research into what you want to do with your PhD?
I think I'm going to do kind of a different project for the PhD. We're still talking it through, so I'm not going to say too much because I don't know how much is common knowledge. I think I am going to do a slightly different thing, but kind of looking at the nearshore stuff.
I don't know.
Here we have cunner.
Whoa!